I'm Day, and this is Microbiology 285L, and I'm going to show you how to do aseptic technique, transfer, and stripping for isolation. We want to focus on doing the aseptic technique because we don't want to have bacteria escaping into the world that we're working in, and also we don't want to contaminate the plates that we're using or the broths. So the first thing we're going to do is turn on our Bunsen burner. Awesome. Like the first try. So important thing to do is sterilizing your loop. And what's great about these is they'll turn super red when they're hot enough to kill anything that can possibly be on it. Awesome. Now, with the Bunsen burner, you have this zone of clearance. So as the air travels through, it's going to kill everything that's around. And the first thing we're going to do is I'm going to do a bacillus plate, and we're going to transfer the bacillus into broth. So I'm sterilizing one more time since I've been sitting here. You don't want to immediately heat this up and then stick it on the plate, because then you're going to kill your bacteria and you won't have anything in your broth. So one good way I like to use is I like to stab the auger in a place where it's not going to be touching anything, and that's going to cool it off. What we want to do is, when we're transferring from plate to broth, you want to grab a colony. Now with bacillus, it's kind of hard to see an actual colony, but I'm going to say this is a colony right here, and I'm going to grab a loopful without gouging into this auger, and you can see it on the loop. And then on my pinky to open, heat up the mouth. Try to knock that down in there. Heat up the mouth once more. And then sterilize my loop. Awesome. Now we're going to plate Serratia, which is this awesome red color, onto this lovely LV plate, and we want to streak for isolation, meaning we want to have little separated colonies everywhere. This is also, if you have any contamination, you will be able to see it. That's another reason why I want to do an aseptic technique. So I'm going to flame my loop, and by using the same technique as I did with the broth, hold your tube in one hand, and you're going to use your pinky to take the lid off, and then heat the mouth, and then to test it on the auger, and then grab, you don't want to grab too much bacteria, so I just tap, probably too much bacteria, and that. Now with the streaking for isolation, you want to place the bacteria like a dot on the edge of the auger, and you're going to spread it out probably a good, I don't know, quarter of the plate, and then you're going to turn the plate, flame your loop, and then you touch once, twice, and then you don't want to touch again because you're going to grab too much bacteria. Flame your loop, turn it again, and do the same process. So I have my bacteria, I'm in zone. I tap my bacteria down and just spread it out lightly because you don't want to cut into the agar. Find the loop, let it cool. And then I turn my flame, and I want to make sure it's not too hot. And then I'm going to touch 1, 2, and then zigzag. Flame one more time. Turn my plate. You want to spread out the bacteria so you only have one bacteria in one little area and it can grow into a nice colony. And then dry. And then that's it. So I did this Monday and today's Thursday, so we should definitely have enough growth. So I'll post a picture, but the problem with this one was that either I didn't have enough inoculum or when I flamed my loop, I didn't grab enough when I went for my second streak. Because you can see that like my initial streak I passed through a couple of times. So usually that's good enough. Sometimes you just don't pick up enough. But I have streaks that I did for another class that actually came out pretty nice. So we have this one right here, which you can barely see, which I'll post, we'll put a picture up for you to see. On this orange one, there's a lot of colonies. There's a lot of colonies that are right next to each other, so it's not the best one, but it's better than having less. And then this teal color one is probably the better one to choose from. Again, we'll put a picture up so you could see it. You can actually see a lot more isolated colonies, so you can have more to choose from, which is always better than having less to choose from. But let's hope that, I don't know. If you guys also want to see a blue one that I did, which is also really cool. With more practice, you get better and better at streaks, and yeah, it's important to get these good so that way, one, if you do have contamination, you can kind of sort out the bacteria that you want versus the bacteria that you don't want. But yeah, so we have an okay streak. I mean, I have 1, 2, 3, 4, 5, 6, 5. I'm gonna say 5. I have 5 colonies that I could pick from, which I mean is okay if I'm only doing 1 or 2 tests with this particular bacteria. But you definitely want more. I don't know if it's easier to see. You want a lot more. And you don't want them touching. You want to spread out the bacteria as thinly as possible because one bacteria is going to be your colony forming unit, so your CFU, and that's what's going to develop into the colonies that you see on the plate.